Hi, everybody. Welcome to this screencast. As I said this past week, that I'm going to be screencasting mitosis and meiosis as well. So this first section will be on mitosis, also known as the cell's life cycle. I'm sure many of you have taken this before. It's sort of integral to most biology courses in high school and some university as well. So for many of you, this will likely be a review. We're not going to go too deep in it, but we need to sort of go through the whole process of what happens. So when we look at the life cycle, there are two parts to it. There is interphase within that life cycle, and then there's what's called mitosis cytokinesis. Cytokinesis is basically defined as changes that occur in the cytoplasm when the cell is under division or about to divide. The only cells that don't follow this life cycle process or this cytokinesis mitosis situation are what we call the sex cells, which are the gonadal cells, as well as the other name, which are the immature sperm for the male, and then of course the ovum for the female. Now, different cells do have this varying life cycle durations. When we look at the you've probably seen it before, the sort of the circle or the cycle, and sort of put in a 24-hour period. So different cells do have different life cycle durations. Now, epithelial cells, which we haven't really talked about, but that's your skin and the mucous membranes in your mouth and so forth. They have a they go through a life cycle very quickly. So we say that they have a lifespan of only a few days. But when we compare it to a neuron, which a neuron is a cell that we find either in the spinal cord or the brain, what you get is what you get inside mom. We don't replace those. So the number of neurons that you have in your brain and your spinal cord, those are set when you're developing inside mom, and you maintain those nerves for the rest of your life. So this is why we say that a neuron can last several decades, because it doesn't get replaced. So this is why something like a spinal cord injury and other nerve injuries or diseases, we can't expect to help them much at all. And there's another reason for spinal cord injuries, but part of it is the fact that these cells do not turn over. They don't go through a life cycle, and once they're gone, they're gone. And this is also the problem we have with alcohol abuse, drug abuse, those sorts of things. These lifestyle situations actually kill neurons, and they don't get replaced. Now, we have lots of extra neurons, but of course, if someone is significantly involved in things like drug abuse, those sorts of things, you can get to a point where we sort of reach a critical mass where we start losing so many neurons that we start losing neurological function, and therefore, we start to see it in our attitude, in the patient's attitudes, their memory, and all those sorts of things. When we look at the human body in its normal change of cells dying and being replaced and so forth, in any given 24-hour period, you lose billions of cells every day from all over your body. So obviously, if we're losing billions of cells, we need to be replacing those cells with new viable cells at any given time. So we have to keep that in mind that, again, when we're looking at, for example, epithelial cells, they're constantly being replaced as they're dying off. When we look at the life cycle of a cell, we actually use what's called a 24-hour life cycle, which compares the various stages. So maybe you've seen this before. This is a relatively common sort of diagram that's used when we're explaining this cell life cycle and mitosis cytokinesis. Right off the top, what I want you to see here in this small little cuts within this circle, we do find mitosis and cytokinesis, but when we compare it to the entire time frame within this 24-hour representation, it's actually a very small part of the life cycle of a cell. So let's do the first part first, and this is known as the interphase. A cell, when it's in the interphase, is not undergoing cellular division. So interphase is the life cycle where the cell is viable, but it's not preparing at all to divide. So when we look at it from, again, this 24-hour cycle of this circle, interphase represents 90 to 95% of any cell's life cycle. So it's not dividing, it's just hanging out under having normal metabolic processes occurring in it. It's a viable cell. But when a cell decides it wants to divide, or right after it's divided, we do break this interphase down into larger parts. And the first is known as growth 1. So when a cell divides into two daughter cells we call them, those two cells that are created out of cellular division, they need to grow and become more mature and therefore viable. So growth 1 represents growth that occurs directly after mitosis cytokinesis. So when the cell divides into two, those two cells do have to go and grow and become more viable. So we say that in growth 1, the cell undergoes significant growth. It also produces the substances that will be required if the cell enters into another division. So you can imagine that, if we look at the creation of the fetus, so when the ovum and the sperm get together and we start this process of creating an individual, of course, we have cells dividing at an unbelievably quick rate to eventually create the fetus, and then the fetus matures with, again, more cellular growth to eventually be born into the baby. So because this happens over and over again, and because even if we look at epithelial tissue that has constant new cells created because of that, when a cell goes into growth 1, it also, if it's going to divide again, needs to produce substances that will be required if the cell is going to divide yet again. So we say that also that this growth 1 is usually the longest stage, whether the life is long or short. So when we look at a neuron, for example, it's in growth 1 after it's created for its entire life. So we say here, a spinal nerve cell can remain in growth 1 for 80 years. 80 years sort of representing a normal lifespan for an individual. So again, we don't replace them. So once they're created, they live with you for 80 plus years. When we look at it from that 24-hour clock here, it represents sort of 8 to 10 hours in its representation of percentage of this whole 24-hour cycle. So we can see G1 here. The next is called synthesis. So if a cell is going to continue into division, some very important things need to happen. The very first thing, if the cell is going to divide, the DNA molecules need to double. The key thing to remember is that when a cell divides and creates two daughter cells in mitosis, those two cells need to have a full complement of DNA material, chromosomes. So because we're going to go from one cell to two, we have to double up the DNA so that both cells that are created in division have viable and the correct amount of DNA molecules to be normal cells. So the first thing that happens in synthesis, then the DNA molecules double, as do the centrosomes and centrioles. They're going to go on to modify into something called the mitotic spindle, but we'll get into those a little bit later. So DNA molecules double, so too do centrosomes and centrioles. The cell, of course, is preparing for cellular division by having this doubling of genetic material. And again, this is represented as 6 to 8 hours of the 24. So we had here, of course, in interphase, we had 8 to 10 hours here, and then the cell is going to divide. It's sort of represented as 6 to 8 in the 24 that we see here. So we've had DNA material double. We've also had centrosomes and centrioles double, and now the cell will move into growth 2. So this is, again, the cell will grow again, but not quite as much as it did in the first phase, growth 1. So in growth 2, we've had the doubling of the DNA material, we've had the doubling of the centrioles and centrosomes, and the cell will continue to grow a little bit more. So growth 2 is the final preparation stage before mitosis and cytokinesis occurs. As we said, it does grow, but not as much as it did in G1, which you remember is what occurs directly after mitosis cytokinesis has occurred. When we look at the representation within the 24 hours, this represents 4 to 6 hours. So when we add it all up at the top end, so 10 here for growth 1, 8 will be 18 hours here for growth 2, and then we've got 4 hours here, we've got 22 or so, or just under 24. So when we look here at the slice of the pie, we see that mitosis cytokinesis only represents a very small percentage of all the things that are happening just prior to when the cell needs to divide. So once we've gone through the three parts here, G1, S, which is DNA synthesis, the doubling up of DNA material, and this little extra growth, we now enter into mitosis and cytokinesis. The definitions of the two terms: mitosis means division of the nucleus, so this doubled up DNA material dividing so that each of the two cells that will come out of division have the appropriate amount of genetic material. And then cytokinesis is defined as division of the cytoplasmic material, which remember is the cytosol, and also organelles that occur in there. And a question some of you may have: yes, you will get DNA replicating itself, so that there's I meant mitochondria doubling up, those sorts of things, so that there will be viable organelles within the two daughter cells. We said this only accounts for about 1 to 2 hours of the 24, so it's a very small percentage in the representation of that cycle. Biologically, though, it actually is the most important stage for reproduction to occur. And when we look at mitosis and cytokinesis, there are 4 phases, and they occur chronologically. First, we have prophase, then we have metaphase, then we have anaphase, then we have telophase. Now, some of you may remember this from school, calling it PMAT, if that's what you remember it as, and that's totally fine. The thing is that one phase does not blend into the other. So there are very significant things that happen in each phase, and they happen, and then the cell goes into the next phase, and other things happen, and then it goes into the next thing, and other things happen, and so on. Okay? So there's a video here that you can watch. I won't put it on here because it doesn't generally tend to come across. You can't hear it well through the microphone that I'm talking in here when I do the presentation. But being said, this is a very good video. It will certainly reinforce what I'm talking about today and help you outside of this screencast as well. So let's talk about the first stage. The first stage is called prophase. We have to remember, when we talked earlier in class, that genetic material in a normal viable cell looks like spaghetti in a bowl. And we did talk a little bit about this when we looked at DNA, when we were talking about protein synthesis and so forth, that we have to take that spaghetti and we have to put it into a viable way in order to have it sort of be able to divide it without any damage or less space. So if you remember, we did have some things that occurred where we start to coil up the chromosomes and the DNA into a certain shape. So we can see here that we had these DNA strands that get all twisted up into these chromosomes. Now you'll notice here that there's 2. Remember, DNA material doubled. So because it doubled, this chromatid is the same as its sister, which is this chromatid. And of course, they'll eventually head off into their separate cells when division occurs. So we have the chromatin, the spaghetti, start to condense and coil, and it will form the chromosomes. And remember that because the DNA doubled during interphase, the chromosomes that appear are formed by 2 chromatids, and they are the same as each other. The 2 chromatids are joined by a central structure known as the centromere. And the centromere kind of holds the 2 of them together. It's an area here that sort of connects them to some degree here, sort of like a bit of a light area where they're sort of stuck a little bit, or they're very close together. When we look at the centromere, we do have this glue called kinetochores and microtubules, and that's sort of holding them together. So when they're created, when the genetic material condenses and coils, it's then held together because it has to move and get put in place before anything occurs, because eventually these are going to get pulled apart, okay? So if we go back again and say, on the outer aspect of the centromere, which is of course this area right here, we find these kinetochores, and these kinetochores are the site for the kinetochore microtubule. So think of this sort of as a little bit of a belt or glue that holds them together, and eventually these kinetochore microtubules will attach to these kinetochores. In the later parts of prophase, the nuclear envelope now disappears. So at this point, we're still in the nucleus. So the nuclear envelope is still there. The DNA material has condensed and coiled. We get the creation of the centromere. And then, of course, then we have the kinetochore, which sort of holds them together. Once that occurs, so we've doubled up the DNA material, condensed and coiled it, right? And then brought the 2 sisters together, connected them at the centromere, and sort of belted them in place with the kinetochores, then the nuclear envelope disappears, as does the nucleolus. So now we have this genetic material that's kind of floating around in the cytosol to some degrees. When we look at these centrioles, they start to migrate at opposite poles. And as the two centrioles migrate, they form these mitotic spindles. Remember the centromeres and centrosomes, or the centrioles and centrosomes. So that's happening that we see here. So we've got, looking at them here, the chromatid, we've got the genetic material that's condensed and coiled. We have them connected at the centromere, and then they're held in place by this kinetochore belt. Then the centrosomes and centrioles, they migrate to either end of the cell to get ready to work with the genetic material, or these chromatids. These mitotic spindles, then they start to create these microtubules. And there are three main microtubules that are associated with the spindle. So again, we look at the picture here, right? We have these microtubules that will eventually connect to the kinetochores, which will help pull them apart eventually. So again, there are three main microtubules associated with the spindle. First, they are the non-kinetochore microtubules. Now they project from the centrioles into the center, but they do not attach to the kinetochores. So again, we have the kinetochores here. So we can see there's a couple of them here in the drawing. So we can see a microtubule here, but it is not attaching to a kinetochore. We can see another one here, not attaching to a kinetochore. So these would be non-kinetochore microtubules. The others are called kinetochore microtubules, and they project from the centriole to the center, and they connect with the kinetochore on both sides. So remember, the centrosomes become the mitotic spindles, and they migrate to the two poles of the cell. They then send out these microtubules. Some do not attach to the kinetochores, and some do. So the kinetochore microtubules, of course, will bury themselves into the kinetochores, so that becomes an attachment at the center area here between the two sister chromatids. So we can see here, we've got the centrosomes here, the mitotic spindle at both ends here, and we can see the chromatids here lined up, starting to line up at the equator, as we call it. So non-kinetochore, they come towards the center of the cell, towards the DNA, the chromatids. They don't attach to the kinetochores. And then we have the kinetochore microtubules, which of course do attach to those kinetochores at the center. Now behind the centrioles, shall we say, we have what are called aster microtubules, and they radiate out from the centrioles, but they don't go towards the center of the cell. They go out behind. And these help form the mitotic spindle, which these help pull the chromatids to the opposite poles of the cell. So we can see here again, we're lining up here. We've got non-kinetochore microtubules. We have kinetochore microtubules here that are into the kinetochores that we can see here in the two chromatids. And then we have these astral kinetochores that come out behind the mitotic spindle. This is a microscopic view, electron microscopic view of a cell. You can see, when we look at drawings here, we see, they look kind of minor. But when we actually look under the microscope, we can see that these are very, very extensive. So here would be the center of the kinetochore and non-kinetochores. And back here would be the astral ones that are sitting out here. And of course, these would be completely surrounded by the cell membrane. The next phase is called metaphase. This is the phase where these mitotic chromosomes, they line up across the mid-region of the cell. So we can see them here. They're starting to line up. So here's our centriole centrosomes, the mitotic spindle. Here, in this case, the artist has drawn kinetochore spindles. They are going to attach here at the kinetochores. And of course, we don't have any non-kinetochores here, but they would be there. And we can see now that the chromatids, the two sisters, they've now lined up along that equatorial line. In this case, this diagram calls it the metaphase plate. I just care about the equatorial line. They're lining up here. So they line up on this equatorial plane. Now, by the time they get to the equatorial plane, the mitotic spindle has grown to its full size, and it's ready to go to the next stage. So we've got the nucleolus gone. We've got these chromatids are connected right at the centrosome and these kinetochores. And then we have the microtubules attached, and we're all set up for the next phase, which is called anaphase. In anaphase, those kinetochore microtubules, along with the asters at the mitotic spindle, they start to pull those chromosomes apart at the centromere. Remember, we had to double up genetic material, and they're all lined up. So we had them all doubled up, and they're all lined up at the equatorial plane, and they're all going to be pulled apart. So they had to each pole. When we look here, we can see they're being pulled to each pole where the mitotic spindles were. Okay, so the chromatids, also known as daughter chromosomes, are drawn to opposite poles by the shortening of the kinetochore microtubules and the lengthening of the non-kinetochore microtubules. It's almost like it's pulled around a pulley. It's like these microtubules kind of use a pulley at the mitotic spindle, and they pull along, and because those kinetochore microtubules are connected at the kinetochores, they yank the two sister chromatids apart from each other so that they're heading to what will eventually become the two sister or the two daughter cells of division. So therefore, because we doubled up and they've been pulled apart, we now have 46 chromatids on one side and 46 chromatids on the other. We have to remember that a viable amount of genetic material to any cell is 23 pair. So therefore, this is why we have 46 chromatids on one side and 46 chromatids on the other. At the latter part of anaphase, the daughter chromatids are almost at either pole, and then we start to create what is known as the cleavage furrow. So you can see that the cleavage is starting here on both sides, and these will eventually come together and pinch off, which will then result in the two daughter cells happening. So we've got the genetic material pulled apart because of the mature mitotic spindle here. The kinetochore microtubules have yanked them apart, and the non-kinetochores have gotten longer as these kinetochores get shorter, pulling the genetic material apart. Once they are pulled apart, we are now into telophase. So now they've been pulled apart. Each pole has 46 chromatids each, which is the appropriate complement of chromatids for a viable cell. And once the chromosomes stop their migration to the poles, telophase begins. And basically what this is, the reversal of prophase. The chromosomes start to uncoil back to chromatin, so they go from the coiled stage back to spaghetti. The nuclear envelope begins to reform around those chromatids as they're returning back to chromatin, and the nucleolus begins to reform. If we remember correctly, the nucleolus was responsible for creating RNA as one of its responsibilities. Sorry, excuse me. So as the genetic material gets pulled apart to the poles, the nuclear envelope begins to form, the nucleolus begins to form, and the mitotic spindles start to disappear because we no longer need to be pulling them around. The cleavage furrow, which started here in anaphase, they start to get closer together. So we say that the cleavage furrow increases until the point that there is now two distinct cells. And now because we have two distinct cells, we say that we now have two identical daughter cells. Now these cells are said to be diploid because they will have two of each chromosome. Pairs 1 to 22, and the 23rd being the sex pair, the XX and the XY, okay? So we can see here now, almost they're together, right? And they're going to continue. We can see the nuclear envelope has started to develop here. The dark purple here represents, we can see the nucleolus is starting to develop, and eventually these chromatids will start to become chromatin, which of course is, as I always refer to, is the spaghetti within the nucleus. Now, we've had the genetic material double up, we've had the genetic material pulled apart at the centromeres, but we have to consider now the organelles that we find inside the cell as well. So we have to remember that, as this cleavage was created, we still had to maintain cytosol and cytoplasmic material, organelles and so forth, in both cells, because they will, once they start to mature, they are responsible for whatever those cells are responsible for. So we say that cytokinesis is the division of the cell cytoplasm, including the organelles. And this process does not begin until the formation of the cleavage furrow, which begins in late anaphase. So as that cleavage furrow gets created and is starting to close up, we see the migration of the appropriate amounts of things within the cytoplasm and the cytosol itself, filling in what eventually will become the two daughter cells. So we can see here that, we've got the breaking down here now of the mitotic spindle. We have chromatin being formed here. We would have the nucleolus being formed, and we can see the nuclear envelope has started to happen. And then we'll see that we maintain a membrane, and we would have migration of organelles and so forth that we would be moving into each one, so that eventually, once we get that full two daughter cells, they have the full complement of DNA, they have a completely functional nucleolus, they have an intact cell membrane, or nuclear envelope. They have any kind of organelles that are required for the function of this cell, and of course mitochondria in there and so forth. Okay, so that ends. Hi everybody, I think I cut myself off there on the last set of slides. I apologize, but that did finish off mitosis. The second part, of course, for this is meiosis, which is a little bit different. So when we think about mitosis, we were in fact actually discussing what are called diploid cells. So this is the creation of normal functioning cells, and diploid meaning that there's two cells that have fully functional and normal genetic material along with all the other functioning things. The other type of division we need to consider is when we want to create reproductive cells. So there's a slight bit of difference that occurs when we're looking at the reproductive cell creation. So in sexual reproduction, each new organism is the result of the union of two different gametes. So when we're looking at mammals, then we have, well, even in some cases, plants and so forth, we do have gametes, which means these are kind of sex cells that used for reproduction. Traditionally sperm in the male, and in the female, they are the ovum. Now, somatic cells through mitosis give us cells with the appropriate number of chromosomes, right? So we don't want the normal number of chromosomes in a cell when we're looking, we do for mitosis, and we want a functional cell that's going to be, whatever the cell is responsible for. But of course, when we're talking about reproductive cells, we are in fact actually needing to consider that when the two get together, when the sperm and ovum get together, they in fact actually each have a half complement of genetic material. So that when the ovum and the sperm get together, then when they connect, we end up with our very first cell that becomes, so as we call it, fertilized. But in actual fact, which is not incorrect, it is a cell that has a full complement of genetic material. So now that we have this very new initial cell of full genetic material, then of course division occurs and we end up creating an individual. So we need to do something to a cell that results in the cell only having half the genetic material required to be a functional cell. So that means when the sperm and ovum get together, they both bring half the complement of required genetic material. And then when the two cells get together, we get a full complement of genetic material. And then this cell can go on to divide and become the fetus and eventually an individual. So we say that meiosis then produces gametes. Gametes sort of represents sex cells with half the number of chromosomes. Because they have half the number of chromosomes, we have to remember that mitosis created diploid cells, two cells with full complement of genetic material. When we look at meiosis and we're creating gametes, we're actually creating what are called haploid cells, meaning that they have one half of the complement of genetic material. Haploid sort of represents single rather than double. So meiosis creates haploid cells, mitosis creates diploid cells. When we have fertilization, of course, as I said, when the ovum and the sperm get together and they fertilize, as the term we use, then we end up with the full complement of chromosomes, as I said, and we can go on to create an individual. So meiosis then is the type of cellular division that occurs in developing sex cells or gametes. In the male, this is known as spermatogenesis. Of course, we think of the terminology with the creation of sperm. And in the female, it is known as oogenesis, which is, of course, the creation of the ovum. And as we say, both of these processes result in the production of haploid cells, only one of each chromosome. In other words, half. We don't have pairs, we just have one. Remember, there's 23 pairs of chromosomes in a normally viable diploid cell. So we can see here that there's a couple things that have to happen. Initially, it looks like normal mitosis, which is kind of correct, but then we have to have something that occurs that results in not getting doubled up genetic material and having cells created that have half the material. So the resulting cells from meiosis are called the sperm and the ovum, respectively. The basic definition for meiosis is one diploid cell, normal number of chromosomes, produces 4 haploid cells, half the complement of chromosomal cells. So whether it's in the male or the female, we do start out with a diploid cell in the areas where these cells are created. Of course, that would be the testes in the male and the ovaries in the female. It always starts out with one cell that divides into 4 haploid cells. Okay, so always the same. Meiotic processes involve 2 nuclear divisions. We have what's called the reduction division, which is known as meiosis 1, and we have the equatorial division, which is known as meiosis 2. So we can see it here. Here is meiosis 1. Here is meiosis 2 over here. So let's talk about those stages and what occurs. We still have a diploid cell that will go under the normal cycle within that 24 hours. G1, which remember is growth directly after mitosis. Synthesis, which is the doubling up of genetic material, and G2, which is a little bit of increased growth. So you're going to say to me, well, I thought you said we create 4 cells with half the complement of genetic material, and that is correct. But before we get there, because we're going to create 4 cells of half material, we still have to create the first 2 cells with full complement. Hopefully you follow my steps here. So 2 cells with full complement will eventually become 4 cells with 1/2. If you do the math, it kind of makes sense. So we still have to go through G1 growth, S, which is synthesis of genetic material or doubling up of genetic material, and G2, which is a little more extra growth. There are still 4 phases in here, just the same as mitosis: prophase, metaphase, anaphase, and telophase. But because we're looking at meiosis, we call it, if we think here, remember, meiosis 1, then this becomes prophase 1, metaphase 1, anaphase 1, and telophase 1. In other words, it's a diploid cell to begin. Again, this doesn't matter whether it's happening in the male or it's happening in the female. You still start with a diploid cell in the process of creating haploid cells. So prophase 1, the nuclear envelope, and then the nucleolus disappear, and the mitotic spindle forms. Same as what we saw in mitosis. It takes a long time for this to happen when we're looking at it in these gametes. So when we look at what's happening here, there is a little bit of difference. So we have to remember, nuclear envelope disappears, or it dissolves, nucleolus disappears, mitotic spindle starts to form. And then what happens is there's some alterations that occur when we compare meiosis to mitosis. When we had the 2 chromatids, so if you remember back in mitosis, we had 2 chromatids, or the 2 sister chromatids, right? They were complete representations of each other on either side, met at the centrosome, remember? Okay, so that was mitosis. In meiosis, those 2 sister chromatids, they pair off, which forms a tetrad. So they pair off, and then parts of those chromatids, they exchange genetic material. So we can see here that we have the 2 sister chromatids coming together. So we have 2 sister chromatodes of each pair. So in mitosis, we just had the 2 sister chromatids, and they lined up at the equatorial plane. In meiosis, the 2 separates, they kind of get together before they get to the equatorial plane, right? So remember, we doubled up, but we set the 2 together. Then what happens is those 2 sister chromatids, they exchange genetic material. So this is known as crossing over. And what results is, because they've exchanged, we end up with what's called genetic recombination. So these were kind of clones of each other, if you want to use that term, but because of this crossing over and the exchange of genetic material, we now have, they're slightly different with each other, which means that, when they get pulled apart, we're going to have genetic material that's slightly different than the diploid cell that originally started in the first place. So again, the two sister chromatids of each pair of the homologous chromosomes, they pair off. This is known as synapsis, and that forms a tetrad, right? 4. 1, 2, 3, 4. Then parts of the chromatids of the two homologous chromosomes may be exchanged with each other, and this is known as genetic crossover, which we see happening here. This is the exchange of chromatid segments, which results in genetic recombination. In other words, it's the formation of new combinations of genes. Okay, so this is how we don't give birth to clones. We actually give birth to individuals because we have this genetic crossover, which means, genetically speaking, the individual that will eventually be created will have genetic similarities, but will be genetically unique to themselves. So imagine that this genetic crossover, we have to remember, I'm talking about both genders here. So this is occurring in the female, in the ovary, within the diploid cell. It's also occurring in the testes in the male, that we have this genetic recombination. So therefore, we suddenly have genetic material that's slightly different. This genetic crossover then allows for 4 genetically different chromatids within the two homologous double chromatid chromosomes. So that means that when they get pulled apart, we're going to have 4 different ones rather than 4 the same. So what happens is, and this is what allows us to be viable and to be to survive as successfully as we have as a species, we get a great genetic variation amongst humans, and this is what has allowed us to be so successful and to survive as long as we have, is because we have this genetic recombination occurring, and therefore such a great variety amongst humans. We still have a lot of similarities, but genetically we're slightly different. Now, because of this, I'll come back to that later. So, at the end of meiosis 1, each cell has 23 chromosomes rather than 46 with mitosis. So in other words, from one cell that is diploid, you produce 4 cells that are genetically different. So here we've got prophase 1, we've got them matching up here into a tetrad. We have genetic recombination between the two, and then they'll start to line up this way and they get pulled accordingly. And you can see here that they're being pulled over, and we'll end up with this genetic material on either side that is slightly different. Now, when we think about mitosis, the double chromatids were pulled apart, right, by the kinetochore microtubules that went to either side. But in meiosis, the double chromatids don't pull apart, but instead 23 go to one and 23 go to the other. And this results into haploid cells. 23 go to one pole, 23 go to the other. So we can see here that, in some ways the diagram here gets a bit confusing, but we get 23 going one way, 23 going the other, rather than 23 pair going one way and 23 pair going the other. So therefore, this results in 2 haploid cells, half the chromosomal complement each. So yes, the genetic material did double up, but they did meet it together. They did have the genetic recombination, and as I said, instead of them being pulled apart to result in the full 46 on each side, 23 go one way and 23 go the other, okay? Meiosis 2 is how we go from 2 haploids to 4 haploids. This also has 4 phases: prophase 2, metaphase 2, anaphase 2, and telophase 2. So we still go through the same steps. But the 23 chromatids from meiosis 1, they do duplicate during the S phase, just like in mitosis. Please remember that we're duplicating half the complement of chromosomes. So back here, because we had 23 go one way and 23 go the other, we are doubling up, but we're doubling up the single amount, so that when they get pulled apart, the 4 cells will still have only half a complement. So formation of 4 haploid cells from 2 is meiosis 2. It still goes through all the same steps, and the 23 chromatids from meiosis 1, they do duplicate during the synthesis phase, but we're only duplicating half the complement of chromosomes. So meiosis 2 follows the same steps as mitosis through all the prophase, metaphase, anaphase, and so forth. And at the end of the process, we end up with 4 daughter cells which contain 23 one-chromatid chromosomes. In the male, spermatogonia is the diploid cell which produces 4 sperms, and in the female, oogonia is the diploid cell which produces 4 ovum, and only one cell is considered viable and the others become polar bodies. So in the male, all 4 sperms do become viable and are included in the ejaculate at any given time. Of course, we have quite a high number of sperm cells that is within the ejaculate, and that allows for the sperm to survive, a percentage to survive to go on to fertilize an egg. Whereas in the female, yes, 4 ovum are created, but only one is considered viable, and the other 4 kind of feed the one that's going to move forward, the ovum that's going to move forward. The polar bodies do provide some nutrition to that ovum as it moves off from the ovary to the fallopian tube and down into the uterus, where things, if it is fertilized, can embed into the tissues of the uterus, and the fetus will start to divide and become a viable offspring. So we can see here telophase 1, right? Then we have it's taken that half amount. Then we have meiosis 2. We did have doubling up of genetic material, but remember, we were only doubling up half the amount of genetic material. Please also remember that we had this genetic recombination that occurred. You can see it here. We have some red with blue and some blue with red and so forth. And then we do have it lining up at the poles. We do have mitotic spindles that do pull them apart at the centrosome. But again, it was only half a complement of genetic material, which results in 4 viable meiotic cells, which can go on to help with reproduction. So in other words, meiosis 1 starts with a diploid and ends with 2 cells each with a haploid number of chromosomes. Meiosis 2, the 2 haploid cells divide with no DNA replication that ends with 4 haploid gametes that are genetically different from the original diploid cell. So you can see it here, right? We've got the genetic recombination happening here, and then we end up with the second part of meiosis, which results in 4 gametes. So this is all I'm going to do. So that means that obviously next week you have Monday off, and then we're coming back after reading week, which is the next week, where we'll be doing your midterm exam, and we'll be starting with tissues, and then after tissues we'll be going into systems, and that's that. So everybody have a great week. I will see you on Thursday, and make sure you do go through this material because some of it will be on the midterm exam in a week or so. Thanks, and have a great weekend.